Okay, so we are in video lecture 2 of 3 for our cell cycle and mitosis playlist, if you will. So at this point, before you start this one, you should already know about the different forms that DNA can take because we're going to start keeping track of that DNA when we're talking about the cell cycle here. So in this video, we're going to start with the question, why do cells divide? Why are we talking about this to start with? And then we'll get into the big picture overview of the cell cycle after that. So, this first question of why cells divide. When we think about an organism's growth, that's one obvious place where we would need new cells, right? So, this is a baby goat and it's going to turn into an adult goat. And adult goats are bigger than baby goats. So, they need more cells to make up that larger organism. So, as organisms grow, they'll add on new cells rather than having their existing cells get really, really huge. So, any organism that is growing physically is going to need to make new cells. We also have certain tissues where we would need to replace the cells pretty often. So, the turnover rate might be really high when the cells become damaged or old or if they die. And some examples of this in our bodies are that our skin cells are often just sort of sloughing off. And as they do so, we need to make new skin cells. And the same thing is true for cells that line our intestinal tract. So, this is a cell that would line your intestine, and these cells are exposed to a lot of stuff brushing against them constantly. And because of that, their turnover rate is quite high. So when we're replacing cells, that's another reason when cells would need to divide. And then last but not least, we have asexual reproduction. And I think this is most obvious in the case of single celled organisms, where something like bacteria, they're going to be able to divide just by splitting themselves into two. So, doing cell replication. Um, but it's also the case for some organisms that are a little bit more complex, like this hydra. So it reproduces asexually just by duplicating its cells and then budding off a new little baby hydra, which you can see here. In each of these cases, our goal is to produce daughter cells that are genetically identical to the parent cell. And it's really important that those daughter cells are identical. Um, if we're making, more intestinal cells or more goat cells, we want them to be genetically the same as the cell we started with. So we want them to have the same set of instructions. It's really important that these daughter cells are going to get all of the chromosomes. And none of the chromosomes are going to be missing, but also no extra chromosomes. We want exactly the right number of chromosomes or set of instructions for these daughter cells. Okay, so that's our why. And now let's start to look at how do these cells divide and what's the process that they go through before they divide. So more generally, what does the life cycle of a cell look like in general? So this is a diagram showing what's called the cell cycle. And what the cell cycle is really just the life cycle of a single cell. And typically, we tend to think of it as starting when a cell is formed and then ending when the cell divides. And it might seem a little arbitrary to say that it's a cycle and then talk about a beginning and an end. But if you think about it, if a cell starts here and then ends up dividing, then now each of the daughter cells is going to go through the cell cycle anew, right? So it's still a cycle, but it's just going to restart again every time daughter cells are formed. And that's sort of the end for our original cell once it divides and turns into two new cells. So when we're thinking about a cell's life cycle, there are two main phases that I want to draw your attention to. And one phase, which is shown here in purple, is called interphase. And interphase encompasses every part of a cell's life cycle where it isn't actively dividing. So if a cell is just growing, um, when it's sort of doing its own thing, all of that's called interphase. And interphase, as you can see here, has three main parts that make it up. So two of these parts are called G1 and G2. And the G1 and the G2 phases are the cell just kind of growing, the cells living its best life, it's doing its own thing. So the cell is maybe making things, it's building molecules, it's carrying out whatever function it needs to carry out. So if it's a skin cell, it's acting as a barrier. If it's an intestinal cell, it's taking up nutrients. So it's just doing its cell stuff in G1 and G2. And then in the S phase, which is the third segment of interphase, this one is really important because it's when the DNA is replicated. We talked in the first video about how DNA can be either replicated or unreplicated. And it's in this S phase, which stands for synthesis phase. So, synthesis, S phase. It's in this S phase when the DNA is copied and it goes from being unreplicated to replicated. And then maybe looking at this diagram, it could be becoming a little bit more clear that this cell section or this cell cycle is going to have a section in it where the cell is going to divide and turn into two new cells. And in order to do that, we needed to double our genetic information first, and that's what happened here in the S phase. Okay, so in interphase, we've got G1 followed by S, where the DNA is copied, and then followed by G2. And then the other main part of the cell cycle, other than interphase, is called the M phase, and that stands for mitotic phase. And this is when the actual chromosomes are divided up between the daughter cells. So, this is the actual division of our genetic material. So, even though we copied our DNA in the S phase, the actual division of the chromosomes between two daughter cells is going to happen in the mitotic phase. And then lastly, one quick little note. So there are some cells that don't actually divide. A lot of neurons in your brain, a lot of retinal cells, a lot of cells in your inner ear. These are cells that once they're formed, they don't really die, but they also don't divide. So some cells are just going to sort of stick around, and they sort of stay in the G1 and they sort of exit it there. We call it the G0 phase. But these are for cells that aren't actively dividing. Okay, so let's think about these different stages of the cell cycle in terms of what the DNA is looking like in each stage. So here is a picture of how we can think of the DNA at these different points in the cell cycle. And we'll start over here in G1, and we'll think of that as our starting place. And you can see that in G1, we've got four pieces of DNA. So one, two, three, four. But none of the molecules are replicated yet. They're all just single pieces. So they're originals, unreplicated. And as we move over into the S phase, this is where we're going to replicate our DNA. And so now you can see that instead of being composed of just a single squiggle, the DNA pieces all have two copies, two pieces to them. So each one consists of two copies, but we still have four chromosomes. We don't count each sister chromatid separately, we count them together because they're just copies. So we still have four, but each of one of the chromosomes we would now say is replicated. And all through this phase, G1, S, and G2, all throughout interphase, the DNA is going to be in that chromatin form where it's unwound and loosely packed. We said it was like spaghetti. So G1, S, and G2 all going to be chromatin, and we're going to be unreplicated. And then starting at the S and into G2, we're going to be replicated. But at the beginning of mitosis, at the beginning of the M phase, it's going to condense into our chromosome form, which is our tightly packed DNA. So it's still going to be in sister chromatids because we're still replicated. And then eventually, as we go through mitosis and we start to sort of divvy up the DNA into two separate daughter cells, we're going to pull apart those sister chromatids so that each daughter cell ends up with exactly the same amount of DNA that our parent cell started with. Okay, so we've talked about G1, S, and G2, and I just want to give you a super, super big picture overview of the stages of mitosis. And then in our last and final video, we'll go into much more detail about the particular stages of what's involved with mitosis. And one note before I name the phases, I just want to say that mitosis is all about the division of the genetic material. So it's all about, we're thinking about DNA. And so when we are talking about the stages, what we're really paying attention to is what the chromosomes are doing and how they're being organized and how they're being portioned out to the daughter cells. So, obviously, in order for this to happen, there are things that are happening with the cytoplasm and the cell membrane and other parts of the cell. But what we're really paying attention to is what's happening with the DNA. Okay, so I cut off the beginning of this diagram, which would have started with interphase because we already sort of talked about that. So we're just starting with the M phase, the mitotic phase. And our first stage here is called prophase, and you can see our chromosomes here. And I'm just sort of naming the stages here, and we'll talk about them more later. So we start with prophase, and then you can see that in this next stage, which is metaphase, our chromosomes are moving to the middle of the cell, and then next comes anaphase, and that's when our sister chromatids are getting pulled apart from one another. So now we just have individual chromosomes at this point. And then the last phase of mitosis is called telophase. And that's where we're sort of reforming the genetic material into the form of two separate daughter cells with two individual nuclei. Okay, so I know, like I said, this description was really, really broad, and you'll have to watch the next video for our explanations of what's going on in each of these stages. So I will see you then.